Welcome to G Fun Facts Online. Imagine a world where medicine isn't just a pill you swallow, but a living, intelligent system navigating your body. Picture microscopic doctors, built from beneficial bacteria or even your own cells, programmed to hunt down cancer, reverse metabolic disorders, and fight off infections with pinpoint precision. This isn't science fiction. It's the dawn of a new era in medicine called Engineered Living Therapeutics, or ELTs. For centuries, our medicines have been external chemicals that interact with our bodies from the outside. They've saved countless lives, but they often come with side effects and a one-size-fits-all approach. ELTs flip the script. They harness the very essence of life, the cell, and program it to become the ultimate therapeutic agent, a living medicine that works from the inside out. Today, we're diving deep into how these cellular robots are built, how they're programmed, and how they promise to revolutionize human health. The power of these living therapeutics lies in their foundation, the cell itself. Scientists aren't creating life from scratch. They're hijacking the incredible machinery of existing organisms. The choice of which cell to use, called the cellular chassis, is critical. Let's start with the smallest workhorses, bacteria. We have a vast community of microbes living in our gut, and scientists are learning to speak their language. Probiotic bacteria, which are already considered safe for us to eat, are a perfect starting point. A star player is a non-pathogenic strain of E. coli called Nissl 1917. Its genetics are well understood, making it relatively easy to engineer. A fantastic example of its use is in treating a rare genetic disorder called phenylketonuria, or PKU. People with PKU can't break down an amino acid called phenylalanine, which then builds up to toxic levels and causes severe neurological damage. The current treatment is a lifelong, incredibly restrictive diet, but researchers have engineered this E. coli strain to produce an enzyme that breaks down phenylalanine right in the gut before it can be absorbed into the bloodstream. These engineered microbes are now in clinical trials, offering a potential future where a living therapeutic could replace this difficult diet. Bacteria are also being turned into tiny cancer fighters. Solid tumors create environments with low oxygen, which certain bacteria are naturally drawn to. Scientists are exploiting this. They've engineered E. coli to home in on tumors and act as tiny, on-site drug factories, producing chemotherapy agents right where they're needed. This minimizes the brutal side effects of traditional chemo that affects the whole body. In another brilliant approach, bacteria are engineered to light up cancer cells, making them visible to the immune system, and then guide our own immune cells to attack. Another safe bacterium being used is Lactococcus lactis, a key ingredient in cheese and buttermilk. It's great for delivering therapies to the gut lining. For inflammatory bowel disease, or IBD, L. lactis has been engineered to produce and secrete an anti-inflammatory molecule called Illinois 10 directly at the site of inflammation, offering a targeted treatment without systemic side effects. Of course, bacteria aren't the only option. The same yeast that gives us bread and beer, Saccharomyces cerevisiae, is also a versatile platform. Because it's a eukaryote, like our own cells, it can produce more complex human proteins, making it a powerful little factory. But perhaps the most powerful living therapeutics are made by reprogramming our very own cells. This brings us to the biggest success story in the field so far: CAR T-cell therapy. This is a revolutionary cancer treatment. It starts by taking a patient's own T-cells, which are a type of white blood cell and a key part of our immune system. In the lab, these T-cells are genetically engineered to express a new synthetic receptor on their surface, called a chimeric antigen receptor, or CAR. This CAR is specifically designed to recognize a protein found only on the patient's cancer cells. Once these engineered CAR T cells are infused back into the patient, they become a living drug. They patrol the body, and when they find a cancer cell, the CAR locks on, activating the T cell and turning it into a potent killer. These activated CAR T cells then multiply, creating an army of cancer-destroying assassins. This approach has led to incredible, lasting remissions for patients with advanced blood cancers like leukemia and lymphoma, who had exhausted all other options. Several CAR T therapies are now FDA approved. So we have the cells, the chassis, but how exactly do we program them to perform these amazing tasks? This is the domain of synthetic biology, which applies engineering principles to biological systems. Scientists are now writing new genetic code to give cells entirely new functions. The key tool that made this possible is CRISPR. You've probably heard of it. CRISPR-Cas9 acts like a pair of molecular scissors, guided by a small piece of RNA. This guide RNA is programmed to match a specific DNA sequence in the cell's genome. It directs the Cas9 enzyme to that exact spot where it makes a cut. The cell's natural repair system then kicks in, and scientists can use this process to edit the gene, or, more importantly, to insert a whole new block of code, a synthetic genetic circuit. This is how we install the new program into the cell. But these programs aren't just simple on-off switches. Scientists are building sophisticated genetic circuits that function like electronic circuits, allowing cells to make decisions. These circuits typically have three parts. First is the sensor. This is the input module that detects a specific signal, like a molecule that only appears when a disease is present. For example, it could be a protein shed by a cancer cell, or the low oxygen environment inside a tumor. Second is the processor. This is where the logic happens. The processor can be designed to perform computational operations. A fantastic example is an AND gate. A CAR T cell could be designed with an AND gate, programming it to attack only when it senses two different proteins on a cell's surface. This dramatically increases safety, as it prevents the T cell from attacking a healthy cell that might happen to have just one of those proteins. Another type is a NOT gate, which acts as a safety switch. The T cell could be programmed to recognize a healthy cell protein, and a NOT gate would keep its killing function turned off as long as that healthy signal is present. The third part of the circuit is the actuator. This is the output. Once the sensor detects the signal and the processor makes its decision, the actuator carries out the therapeutic action. This could be producing insulin, releasing an anti-inflammatory drug, or in the case of CAR T cells, activating the cell's killing machinery. This technology is incredible, but creating a living medicine is not without its challenges. These are major hurdles that scientists, engineers, and regulators are working hard to overcome. First, there's the problem of stability. Living cells divide and evolve. There's a risk that the carefully designed genetic circuit could mutate over time and stop working, or worse, evolve in an unpredictable way. Ensuring these programs remain stable for the long term is a huge engineering challenge. Then there's cost and scalability. CAR T therapy, as amazing as it is incredibly expensive, costing hundreds of thousands of dollars per patient. The process is complex. A patient's cells are harvested, shipped to a special facility, engineered, grown, and then shipped back. Making this process more automated and affordable is essential to make these therapies widely available. But perhaps the biggest concern is safety. We're putting a genetically modified organism into the human body. What if it escapes its intended location? What if it's shed into the environment? To prevent this, scientists are designing ingenious biocontainment systems, often called kill switches. A dead man switch, for example, programs the cell to require a special synthetic molecule to survive, a molecule not found in nature. If the cell escapes the controlled environment where this molecule is provided, it can't repress a lethal toxin, and it self-destructs. Another type is a temperature-sensitive switch. A cryodeath switch is designed for microbes meant to work at human body temperature. The circuit activates a lethal toxin if the temperature drops to that of the outside environment. ensuring the microbes die if they are excreted from the body. These safety measures are absolutely critical for gaining regulatory approval and public trust. The U.S. Food and Drug Administration, or FDA, is actively developing frameworks to oversee these products, which don't fit neatly into the old categories of drugs or devices. And this brings us to the ethical questions. The power to reprogram life is profound. We must ensure the safety of patients, address concerns about playing God, and, crucially, tackle the issue of equity. If these life-saving treatments are only available to the wealthy, we risk creating an even wider gap in health disparities. So where does this field stand today? And what's next? This isn't just theory anymore. Engineered Living Therapeutics are moving rapidly into clinical trials. While CAR-T therapy is already approved, the next wave is focused on engineered microbes. A company called Synlogic is developing engineered E. coli to treat PKU, the metabolic disorder we talked about earlier. And their work is in clinical trials, providing vital data on the safety and efficacy of these living medicines in humans. And the future is even more exciting. We're moving towards cells controlled by more complex logic, integrating multiple disease signals at once. Imagine a CAR-T cell that only attacks a tumor if it detects 2 cancer proteins, and a signal of local inflammation, all while being controlled by an external drug the doctor can switch on or off. We're also seeing the rise of theranostics, integrated systems that both diagnose and treat disease. An engineered microbe could patrol your gut, sense the earliest molecular signs of colon cancer, record that information for a later diagnosis, and simultaneously release a small dose of a drug to nip the problem in the bud. Soon, we might not use single strains of bacteria, but engineered cocktails or consortia, where different microbes work together to perform complex tasks, creating a synergistic therapeutic ecosystem inside our bodies. The line between the digital and biological worlds is also blurring. Techniques like optogenetics, which uses light to control cell activity, could allow doctors to adjust a patient's internal living pharmacy in real time, using external signals, perhaps from a smart wearable device. And designing these incredibly complex biological circuits is a monumental task. This is where artificial intelligence comes in. AI and machine learning are becoming essential tools to model, predict, and design the next generation of ELTs, dramatically speeding up development. Engineered Living Therapeutics represent a fundamental paradigm shift. We are moving from static, chemical interventions to dynamic, biological collaboration. By programming the machinery of the cell, we are creating a new class of medicine that is intelligent, adaptable, and deeply personalized. The challenges are real, but the potential is undeniable. From our own supercharged immune cells eradicating tumors, to engineered bacteria silently rebalancing our metabolism, we are just at the beginning of what is possible. The microscopic doctors are in, and they are ready to get to work.